Thank you for inviting me for this wonderful virtual meeting. My name is Peng Liu. I'm from University of Minnesota. The title of my talk is Molecular Implication of a Caspase-2-Mediated Site-Specific Tau Cleavage in Neurodegenerative Disorders. I have nothing to disclose. The microtubule-associated protein Tau is a highly soluble protein enriched in neurons. In a neuron, Tau mainly reside in axons, but a minor portion reside also in somatodendrites and synapses. In addition, in the central nervous system, alternative splicing of Tau message RNA result in 6 different splicing isoforms of Tau differing in amino terminal region and the microtubule binding domain. In addition, Tau is subjected to post-translational modification. There are at least a dozen types of different modifications have been identified, including truncation and the most prominent phosphorylation. Such modification, together with alternative splicing and subcellular location, confers upon Tau its tremendous heterogeneity, which subserves its many physiological and pathophysiological function. Under normal conditions, Tau is actively involved in a variety of processes. However, under disease condition, the carefully choreographic Tau modulation go awry, forming toxic in both neuronal cells and glial cells, and forming insoluble inclusions, including neurofibrillary tangles. Such inclusions are pathological hallmarks of a group of neurodegenerative disorders, collectively known as Tauopathies. There are a total of more than 20 Tauopathies has been identified so far, and the family of Tauopathies is growing. Tauopathies can be classified into primary Tauopathies, secondary Tauopathies, and some geographically isolated forms. In primary Tauopathies, such as Lewy body dementia and FTDP17, phospho-Tau aggregate is the main pathological finding. In secondary Tauopathies, such as Alzheimer's disease and Huntington's disease, some other protein deposits occur. The 3 main clinical manifestations of Tauopathies are motor dysfunction, cognitive decline, and changes in mood. As cognitive decline is common among Tauopathies, a key question to ask is what Tau forms cause cognitive decline? Early clinical pathological studies suggest that neurofibrillary tangles are associated with cognitive decline. However, the causative role of NFTs was challenged by later findings from human brain studies and studies using models of Tauopathies. Particularly, several independent studies from groups unanimously showing that synaptic dysfunction and cognitive deficits is temporarily deassociated from NFT, and the memory impairment can be ameliorated even in the presence of NFTs. So such finding rather suggests that soluble Tau forms trigger cognitive impairment. So in this talk, I'm going to share with you our identification and characterization of cognition-related Tau molecule called delta Tau 314. I'm going to talk about the relevance of delta Tau 314 to various Tauopathies. And then we're going to discuss the impact of this cleavage event on synaptic transmission and cognitive function. So we use RTG4510 mice to characterize and identify and characterize soluble brain-derived proteins that affecting cognition. These mice developed cognitive deficits before mature neurofibrillary tangle and neurodegeneration occurs. We assess the spatial reference memory of the mice using Morris water maze at 3 months of age. Meanwhile, we thoroughly examined the brain-derived soluble tau proteins, and we identified a 35 kilodalton tau protein, TCP35 for short, which the level of TCP35 is higher in mice which manifest cognitive impairment than those cognitively normal. Meanwhile, we show that level of TCP35 is inversely correlated with memory function of mice. This tau protein is a C-terminally truncated tau because it can be detected by antibody tau13, which recognizes the N-terminal region of tau, but not by antibody tau46, which recognizes the C-terminal region of tau. Mass spectrometry analysis of this protein purified from the brain of RTG4510 indicate that its C-terminus ends at aspartic 314, hence the name delta tau 314. In vitro fibrillation and sedimentation assay using recombinant protein suggest that delta tau 314 can form thiofilament T reactive fibrils less readily and precipitate to a less extent than their full-length tau precursor. This is likely due to a near complete elimination of tau aggregation core that spans between valine 306 and phenylalanine 378. Proteases cleaves after aspartate includes caspases, matrix metalloprotease, and granzyme B. Based on the sequence flanking aspartate 314, the strongest candidate for tau hydrolysis is members of a caspase family. We perform in vitro cleavage assay and show that among the 8 caspase that expressed in human central nervous system, caspase-2 is the only one that catalyzes the formation of the delta tau 314. And importantly, delta tau aspartate 314, when mutated to glutamate, it can block caspase-2 catalyzed delta tau 314 production. So far, I showed you that delta tau 314 is a C-terminally truncated tau fragment produced by caspase-2 catalyzed tau cleavage at D314, and is less prone to fibrilize than full-length tau. So is delta tau 314 relevant to tauopathies? To answer that question, we first asked whether delta tau 314 is present in human brains. Using immunopurification coupled Western blot, we show that there are multiple protein bands present when detected by delta tau 314 specific antibody. Mass spectrometry results confirm that the identity of these bands are delta tau 314 that is derived from different tau isoforms with 4 or 3 microtubule binding domain. In collaboration with Mayo Clinic at Rochester, we studied delta tau 314 in elderly individuals with AD-type dementia, mild cognitive impairment, and cognitively normal. This group are comparable in sex ratio and postmortem interval. IP Western results and ultrasensitive Simoa results consistently show that level of delta tau 314 is higher in cognitively impaired, which combine AD dementia and MCI, than cognitively normal. In addition, level of Caspase-2 is also increased in the cognitively impaired group. Moreover, level of delta tau 314 modestly predict cognitive impairment, although not as effective as amyloid and tau pathology, hallmarks of AD. In collaboration with University of Washington, we studied delta tau 314 in Lewy body dementia, in which there are two groups. One is Parkinson's disease with dementia and Parkinson's disease without dementia. These two groups are comparable in age, sex ratio, and postmortem interval. We show that delta tau 314 is increased in Parkinson's disease with dementia compared to the group without dementia. In addition, levels of Caspase-2 is also increased in the dementia group. Delta tau 314 can predict cognitive impairment as effective as Lewy body staging. We also acquired brain tissue from NIH Neurobiobank and New York Brain Bank to study delta tau 314 in Huntington's disease. We show here that in prefrontal cortex, the level of delta tau 314 in HD is higher than that in non-HD, and level of Caspase-2 is higher in HD as well. And level of delta tau 314 and Caspase-2 correlates. And the same finding apply to the case of brain tissue of striatum. So I hope you can appreciate that delta tau 314 is associated with multiple tauopathies. So what's the relationship between Caspase-2-mediated tau cleavage at D314 and synaptic transmission? Well, under normal conditions, as I mentioned before, a minor portion of tau reside in dendrites and dendritic spines. However, when cells are challenged by stress stimulators such as A-beta oligomer related to Alzheimer's disease or expressing P301L variants associated with frontotemporal dementia, tau is hyperphosphorylated. And this hyperphosphorylated tau is dissociated from microtubule, enters to dendritic spines, and accumulates the dendritic spines, which leads to internalization of glutamatergic NMDA receptor and Impair receptor, and in turn reduce synaptic transmission. As you can see, tau abnormal accumulation in dendritic spines represent an early neuropathological changes in synaptic function. What remains unclear is what drives hyperphosphorylated tau to dendritic spines. To answer that question, we use in vitro primary neuronal culture. Different tau variants were transfected into neurons and expressed as EGFP tau fusion protein, and DS-RAD was used to show neuronal morphology. Consistent with what we previously found is that expressing tau P301L leads to tau accumulation to dendritic spines, whereas expressing wild-type tau doesn't. However, when a D314E is introduced to tau P301L to block the cleavage, you see no obvious tau in the dendritic spine. Interestingly, the cleavage product, delta tau 314, can accumulate to dendritic spine. Notably, expressing different tau variants leads to no alteration in the total number of spines. This data suggests that cleavage at D314E is required for tau to accumulate in dendritic spines. Next, we address whether cleavage at D314E triggers tau at hyperphosphorylated state to accumulate to dendritic spines. In tau, there are 14 serine and threonine residues reside immediately before a proline residue. And these residues can be phosphorylated by proline-directed kinases. And hyperphosphorylation of these residues has been shown to modulate neurotoxicity in fruit fly. Here, we mutate all 14 residues to alanine. The resulting AP constructs block tau phosphorylation. In parallel, we mutate these 14 residues to glutamate. The resulting EP constructs mimics tau hyperphosphorylation. Expressing EP leads to tau accumulation to dendritic spines. However, when a D314 is introduced to block the cleavage, tau accumulation to dendritic spines is no more. Interestingly, expressing AP do not lead to obvious tau mislocalization to dendritic spines. Despite that, the D314 cleavage site is not blocked, and that delta tau 314 derived from AP construct can accumulate to dendritic spines. This data suggests that both phosphorylation and cleavage at D314 are required for tau to accumulate to dendritic spines. In addition, we show that knocking out caspase-2 leads to tau P0L to dendritic spines to a lesser degree, whereas accumulation of delta tau 314 is not affected by caspase-2 genetic ablation, indicating that caspase-2 is required for tau to accumulate to dendritic spines. Next, we study the effect of tau expression on synaptic location of Impair receptor by measuring the overlapping fluorescence intensity between GUT-R subunit of the receptor and PSD95, a scaffold protein enriched in excitatory postsynaptic membrane. We show that compared to neurons expressing tau wild type, expressing tau P301L and delta tau 314 significantly decreased overlapping degree, whereas introducing a D314E to block the cleavage to tau P301L reverse the overlapping degree to a higher level. This suggests that cleavage at D314 is mediate Impair receptor internalization. We studied the synaptic transmission by measuring miniature excitatory postsynaptic current. We show that compared with neurons expressing EGFP only, expressing EGFP tau P301L decrease both the amplitude and frequency of miniature EPSC, whereas such a decreased effect was brought back by additional D314E. This indicates that the P301L expressing causes excitatory synaptic transmission deficits, which can be reversed by blocking tau cleavage at D314. So far, our model support the following, our data support the following model, which is expressing tau P301L triggers cellular stress, which activates protein kinases, leading to tau hyperphosphorylation. Caspase-2 cleave hyperphosphorylated tau to produce delta tau 314. And this cleavage induced dendritic spine accumulation of delta tau 314 and other tau forms, which we will discuss more in the next section. And this abnormal accumulation of tau leads to glutamatergic, such as AMPA receptor internalization, and eventually impairment in synaptic transmission. So how does delta tau produced event affect cognitive function? To answer that question, we first use morpholino antisense oligonucleotides to block caspase-2 expression in RTG4510 mice. As I mentioned before, these mice developed spatial reference memory impairment before neurodegeneration. So we set up the experiments within this period between 3.5 to 5.5 months of age to avoid analyzing deficits due to neurodegeneration. Mice were first test their spatial reference memory using Morris water maze. 2 weeks later, mouse were treated either with morpholino or vehicle for another month. At the second half of the treatment, mouse were retested using Morris water maze, but at this time, the target quadrant was relocated to prevent previous memorizing effect. We show that unlike RTG4510, the non-transgenic TG transgenic mice memorize target quadrant well, regardless of the treatment or the test age. Whereas in RTG4510 mice, the percentage of time when they treated with morpholino, the percentage of time that mouse spent on target quadrant is increased from 25%, implicating no memory function, to about 40%, indicating an improved memory function to the level of non-transgenic mice. Importantly, this restoration of memory function is associated with about 35% decrease in levels of caspase-2 following morpholino treatment compared to the vehicle, and about 35% decrease in delta tau 314 without altering full-length tau level. Next, we asked whether D314E prevents P301L-induced cognitive deficits. To answer this question, we use bilateral intra-hippocampal transduction to wild-type mice using adeno-associated virus carrying EGFP tau fusion constructs. The surgery was performed starting at 2 to 3 months of age, and we perform Morris water maze to test spatial reference memory at 4 to 5 months of age. We show that a longer path length in finding hidden platform in mice expressing EGFP tau P301L than mice expressing EGFP only or EGFP P301L D314E. This suggests that D314 mutation induced blockage of tau cleavage prevent tau P301L induced cognitive deficits. Last, we studied the direct effect of delta tau 314 expression on synaptic and cognitive function. We used the similar experimental paradigm. We observed, however, that mice expressing EGFP delta tau 314 perform no difference than mice expressing EGFP only, indicating no expression delta tau 314 does not disrupt spatial learning or memory. Despite that, the level of delta tau expressed in postsynaptic compartment of these mice is approximately 10 times higher than those in mice expressing tau P301L. We know that the mice expressing tau tau P31L exhibit cognitive impairment. However, noticeably, you see that there are other tau forms, including full-length tau, be present in postsynaptic compartment of P31L expressing mice. In addition, we show that in cultured neurons expressing EGFP tau, delta tau 314 alters neither amplitude nor frequency of miniature EPSC, indicating no detrimental effect to synaptic transmission. So this data indicate that Caspase-2-mediated cleavage tau at D314E can trigger the infiltration of not only delta tau 314, but also other tau forms. It is the latter, I'm sorry. It is the latter, but not the former that triggers impairment in synaptic transmission and cognitive function. So in summary, I showed you today that delta tau 314 is a cognition-related molecule and is associated with various tauopathies, and Caspase-2-mediated tau cleavage leads to synaptic transmission and cognitive dysfunction. Based on our data and studies from structural biology, physiology, and clinical study, we believe that targeting Caspase-2 may be effective for improving synaptic transmission in neurodegenerative disorders. So I'd like to thank colleagues in University of Minnesota, particularly my faculty mentor, Dr. Karen Ash, and external collaborators and funding. Thank you for your attention.